Today we can discuss about the principle behind plasmid isolation. What will be the difference between DNA isolation and plasmid isolation? How we can isolate plasmid from bacteria? This is the one which we are going to discuss. As we know that DNA isolation can be done through phenolchloroform method, plasmid isolation is done by another protocol called alkaline lysis method. So that alkaline lysis method is what which we are going to discuss today in this video. What is the first step for isolation of plasmid from bacteria? The first step will be to grow the bacteria in LB broth containing the selection antibiotic. Usually, the plasmids will have some antibiotic resistant genes in them. So, you can use the what the selection antibiotic which is present in the plasmid so that we can use it for the selection of the bacteria containing the plasmid. So first will be the growing the bacteria in the selection antibiotic. After growing, we will if we get when we get the sufficient amount of growth, we will centrifuge the bacteria so that we will get the bacterial pellet. Next step in the isolation. Next step means the first step. First, the growth of bacteria. The first step in the isolation of plasmid or extraction of plasmid is the this bacteria pellet which we obtain after centrifugation. We will resuspend it in a resuspension buffer. What is this resuspension buffer? It is typically the basic pH tris buffer, tris EDTA buffer, which we use to resuspend the bacterial pellet. What is doing? This tris EDTA buffer destabilizes the cell membrane as well as it inhibit the DNases as well as RNases during the process of plasmid isolation. So this is the first process of in the plasmid isolation. That is a resuspension buffer. The second step is lysis. After resuspension of the cell, destabilizing the cell and inactivating DNases as well as RNases. Now we need to break open the cell. As we seen in the DNA isolation also, it is a break open the cell is the main step because we need to break open the cell, remove the unwanted material so to isolate our material of target. Here also the lysis, but a slight difference from that of the DNA. Here we use alkali for the lysis. That is because of that only we call this method alkaline lysis method. We use NaOH alkali for the lysis as well as SDS. What this SDS is doing? SDS, as we know, it is a detergent. It will remove the phospholipid and it will solubilize the protein so that the cell will break open. The same SDS we use for the DNA solution also, but here also the SDS is doing the same thing, which is removing the phospholipid and solubilizing the proteins. One thing which we added extra apart from the DNA solution, that is the NaOH, that is alkaline. What is NaOH doing? Sodium hydroxide denature a genomic plasmid DNA as well as the protein. Sodium hydroxide, what it is doing is that it denatures the genomic DNA, plasmid DNA as well as proteins. So the genomic DNA, plasmid DNA, and the proteins get denatured. Genomic DNA, plasmid DNA will get converted from the what is single strand, double strand to single strand. You can see here. We can go back to the earlier picture. You can see it was double stranded. The circular DNA as well as genomic DNA. This is a plasmid and this is a genomic DNA. Both were double stranded. But here you can see it becomes single stranded. So denaturation occurred and the protein also get denatured. Cellular DNA become linearized and strands are separated. You can see that the cellular DNA gets separated, but the plasmid DNA also gets separated but the plasmid DNA circular remains topologically constrained, that it is the two strands denatured will not separate, but it to remain together. Here is our here lies our principle. So we have lysed and we have denatured DNA, protein, and DNA, DNA as well as protein. DNA means the genomic DNA as well as plasmid DNA. So then we add neutralization solution or neutralization buffer. The neutralization buffer which contains potassium acetate. What is the role of potassium acetate? It neutralizes. So the addition of potassium acetate results in the high salt concentration, and it will try to neutralize the pH, so that the SDS which we added in the what cleavage, the lysis step, SDS which we added in the lysis step, as well as lipids, the lysis step, the lipids has been removed by the SDS. The proteins get precipitated as a white precipitate by the addition of potassium acetate. So, and neutralization, as we add the neutralization solution, as the solution is neutralized, the DNA, the genomic DNA and plasmid DNA start renaturing. It will start forming the double stranded DNA. As we as this white precipitate is formed, we will take this tube and centrifuge, so that this genomic DNA will not get genomic DNA is a big DNA. It will not get enough time to get come back double strand. But the plasmid DNA, it is already these two circular single strands were held together during the denaturation also. So it will renature faster. So the genomic DNA will try to renature, but it will get captured in this white precipitate and get separated in the debris. While in the solution, there only the plasmid DNA will remain. The genomic DNA will try to renature, but it failed to renature because it is very big DNA. As we are centrifuging, it will get trapped in this white precipitate of SDS, lipid, and protein and get removed in the debris. While in the solution, there only now the plasmid DNA remain. So the precipitate and chromosomal DNA is removed by centrifugation. So the last step, that is the purification or precipitation of them. So our solution containing now the plasmid DNA. What we'll do next, just like the DNA, just like what we do in the DNA solution, our solution does not contain any genomic DNA. We will add isopropanol or ethanol for precipitation. Or else nowadays we use columns. We use silica columns or glass fiber columns for the separation. Both technique can be utilized for the separation of or precipitation. In the first case, it is a precipitation of plasmid DNA. Or in the second case, the plasmid DNA will be immobilized in the column and later eluted, so that we will get the pure form plasmid DNA. So both technique can be used. This is about how we can isolate or how we can extract plasmid DNA from bacteria. It done through alkaline lysis method. It is a bit different from that of the phenochloroform method, which we use for the genomic DNA isolation. Hope you understood the topic. For more videos like this, please subscribe to the channel. Thank you.